Today we have Jonas Coussement, who will give a talk. He is a postdoc in Belgium at Ghent University, working in the lab of Coussement, and he has been working on FSP modeling since more than 10 years, probably.
10 years, yeah.
And today he will talk about water transport modeling.
Yeah, so hi everyone. I thought I'd start this presentation with just a quick rundown of what the pivotal role of water is in plant growth. And one of the first things to know that in a plant, water flows from based on a gradient in water potentials. So water flows from a less negative to a more negative water potential, and this water potential represents sort of a pulling force that is drawing the water towards it. So here you have an equilibrium state during the night, for example, where the leaf water potential is minus 0.05, the stem water potential is the same, and the soil water potential as well. And then the sun comes up, and what happens? The plant starts to transpire. It starts to lose water through the leaves. And this is because the water potential of the air is also much more negative than from the plants. And so due to the water loss in the leaves, the water potential in the leaves starts to decrease. It becomes more negative. And this means that the leaves will start pulling on the water that is contained in the stem, which means water flows from the stem into the leaves. The water potential of the stem drops, and eventually it starts refilling the plant organs from the soil because of this gradient in water potential. Then the sun goes down again, transpiration stops, and it takes a bit of time for the plant to refill all the water that was lost during the day, and eventually we reach our equilibrium state again. That's the simple explanation of how water flows in a plant. Now, what do we notice in, what happens when a plant has a water deficit? For example, on the photosynthesis side, well, both the take-up of CO2 and the transpiration happens through the stomata. And if the plant doesn't have enough water, it closes its stomata, and it also causes a drop in photosynthesis. That's something that we know. But when we look at various plants and really look at the growth processes, we notice that despite the photosynthesis dropping moderately, that the growth drops much faster, even at like moderate levels of drought stress. And so even during these daily fluctuations of water availability, we really have water having a much larger impact than just on photosynthesis alone. So if we look at what's happening in a cell, a normal cell has a vacuole where water flows in and out, just on a normal equilibrium state. And what happens when the plant starts to do photosynthesis and starts creating sugars is that it's going to actively load sugars inside this vacuole. And this causes the osmotic potential to drop. And the osmotic potential basically means that if you add solutes to water, that the water potential will also drop. So it will get more pulling power to draw water in from its surrounding cells. So what will happen is water will be pulled into the cell, which now contains water and additional soluble sugar. And this will exert a force on the cell. So it will exert a positive pressure on the cell walls. And this is called the turgor pressure. And this is what is actually causing the plant cell to grow. So this will cause new plant wall material, cell wall material to be formed and cause irreversible growth. So to look at this a bit more in detail, so if we look at a water potential, we basically, in a plant, we have two main components. So the osmotic potential that I just talked about, which is a negative aspect of the water potential, and the turgor potential, which is a positive aspect. And if you sum both together, you get the water potential, and it's this water potential that I showed earlier that is driving the water flow within the plant. In trees, you have another component, which is the gravitational potential that's also having an impact, but it's only for like very tall trees. And in the soil, you also have another matrix potential. But for plants, we can only focus on the turgor and the osmotic potential. So what happens if we transpire? Our cell loses water. Our osmotic potential will drop. Why? Because the concentration of sugars is increasing in the vacuole. so we're having a more negative osmotic potential, our turgor pressure will also drop because we have less pressure, less external pressure on the cell walls because we're losing water. And this means our total water potential will also drop, which means that the plant will be drawing, well, the plant cell will be drawing in more water from its surroundings. When we add sugars, what happens then? We see the osmotic potential starts to drop, but not necessarily something happening with the turgor pressure yet. This drop in osmotic potential, like I showed earlier, draws in water, so our water potential becomes more negative. But what happens now is that our turgor pressure starts to increase. And when it increases above a certain threshold, this is when the plant cells can actually start growing. So the first part of like the turgor pressure variations are causing an elastic component. So the plant cell can shrink and expand elastically, but once we exceed a certain threshold, it starts creating new cell wall material and it starts growing. And this this principle is captured in a well-known equation that's called the Lockhart equation. And it basically says that a change in plant cell volume, in this case, has an elastic component that is based on the changes in turgor pressure, and it has a plastic component, which is our actual growth, that is based on the turgor pressure as well, but the turgor pressure exceeding this threshold value. And this is the line that is also shown here. So that's generally the concept of like the turgor-driven growth. So what happens now if we apply these concepts to an entire plant and not just to a single cell? And for this, I also included the equation. So I'll try to do it quite slowly to go over it, but it's quite straightforward. So we have here, the first equation is the equation of the osmotic potential. And basically what's important here is that you have the mass of sucrose that is in a plant cell or in a plant organ divided by the volume, the storage volume of the plant organ, which is sort of saying the amount of water that is available in the plant organ. So basically this is the sugar concentration. And the water potential is the sum of the osmotic potential and the turgor pressure. Then the first thing that we're interested in is the flow between organs. So this is the flow into a plant organ, like I said earlier, is described by the difference in water potential. So between an internode and a leaf, it's difference in water potential between the leaf and the internode, divided by a transport resistance. So if you have two organs that are further away from each other, or you have like a tree that is wider, you have less resistance for your water to flow upwards. And this impacts how fast the water can flow within your plant. Yeah.
You shared R in this equation is a different R from the.
Yeah. Good point. Yeah, this first R is the gas constant. Yeah, that's a good point. And this is a resistance factor. Yeah, that's a good point. Yeah. Yeah. And so what we're interested in knowing is exactly how much the water content of each plant organ is changing. So for a leaf, it's quite straightforward. It's the amount of water flowing into the leaf, which we have calculated with our differences in water potential, minus the amount of water leaving the leaf through transpiration. But for our internodes, it's a bit more complicated because we can have water flowing towards one other organ, or it can flow upwards to a second organ, a third organ. So we actually need the difference between the water flowing into the internode minus the sum of all the water that is leaving all the other plant organs that are lying connected above the internode. And then we need to go from our change in water content now to a change in organ volume, which we can do straightforwardly by just assuming that water is cannot be compressed or expand. So the change in volume is directly related to the density of water. So this is basically change in water content is the density of water times the change in plant volume. But then we need to know, is this change in plant volume that we're modeling, is this the elastic change of the Lockhart equation? Is it actual growth? And how do we translate this into 3D for our functional structural plant model? So let's go back to the original Lockhart equation. So this is just a general purpose description of how we should translate volume differences into like the elastic and the plastic component. And the thing that we can do is we can decompose this equation by not just focusing on the volume in itself, but we can decompose it into each cardinal direction. So we can use 3 axes, for example. Instead of looking at the change in volume, we can look at the change only in the z-axis or in the x-axis or in the y-axis by using a different extensibility, this parameter is called, which is a parameter that determines how fast a plant cell can grow in response to its turgor pressure. And so you can have a different extensibility in different directions. So for example, for a round seed, you can say it's like the same extensibility basically in each direction. But for a leaf, it will be much higher in the longitudinal direction than in, for example, the thickness direction, because it grows much less in the thickness. And so now, here's a practical example of how we calculate combine this all to get to a change in turgor pressure for a perfect cylinder. So assuming an internode can be modeled as a perfect cylinder. So we start from, we have the change in water content in our plant organ. And we know this relates to our change in volume, in our plant volume. And then we know the volume of a cylinder can be described as just the length times pi times the radius squared. And so we do, as I said earlier, we decompose this equation, 1 divided by v on dv. This is like just a derivative in terms of its specific dimension components. And so we can use a specific Lockhart equation for each direction. So you have one in the longitudinal direction using the z axis, and you have one in the radial direction, and we are assuming that both radial directions are basically the same. So you have an x component. And now you can do some fun recombination between all these components. So looking at everything that's here in this third equation, you can basically fill in each portion of this equation with other components of the other equations. And then you do some recombination. You can do this in your own time, and you get an equation that looks like this. It looks very complicated, but it's basically expressing the change in turgor pressure in terms of the change in water content and the extensibility and elasticity components. So it allows us to well, estimate or calculate if an amount of water is entering our plant organ, how much will our turgor pressure change? And then we can use this turgor pressure again in these Lockhart equations to calculate the actual growth, both elastic and non-elastic for our plant organ. And so, like I said earlier, if you have different plant organs, like an internode, you will have this extensibility in the z-axis that is much higher than in the radial directions. And then basically, even if you start from a square object, just the water flowing in will have like the elongating effect. And so for a leaf, it's 3 different components. And for like a seed, it's almost, or a fruit like a tomato, they are basically all the same. And then we have the implementation. So I work with GrowImp, but I'm still working on GrowImp 1.6, so I hope it's all still relevant. Also, I'm not sure how it translates to like Julia and MATLAB, but basically the idea is, I think, the same. But I'm showing the specific implementation of how I did it in GrowImp. So what are the main issues that we can have with this implementation? Is that we need to calculate the flow only to and from connected plant organs. So that's what we have GrowImp's graph structure for. We also have the problem that our equation set changes with a changing plant structure. So when we're creating new plant organs, we're constantly adding new parts to our network where we need to calculate the flow in between of it. And then we need to solve the set of differential equations for the entire structure. So starting with the first one, it's in GrowImp, you can basically define a super module that is a plant organ. So Graham knows that both a leaf, an internode, or a fruit are all parts of like a plant organ. And you can basically add to your module every aspect that you need, like the sugar content, the extensibility, the elasticity, and stuff like that. And then you just define your internodes, your leaves as plant organs, which will be useful for later. So we can just define the equations as being two plant organs interacting with each other without having to specify each plant organ individually. Oh, I have this twice apparently. And so how do we calculate the sap flow between two connected organs then in Graham? Well, we basically say we look for each plant organ that is below another plant organ, or yeah, in two directions you can choose. And you basically just take the difference between water potential of two connected plant organs. You divide it by the transport resistance, and you basically get your sap flow into the plant organ. So not out of, but into the plant organ. The second problem we had is that our equation set changes with our changing plant structure. So remember this slide here, that we have this summation of all these plant organs that we need to find. And in GROIMP, this is also quite easily solved with the query function. So basically, if we want to define the change in water content, we take the sap flow into the plant organ, and we subtract the sap flow of all the plant organs that are connected one higher onto this internode with the sum function. So this finds from our internode that we defined all plant organs that are located exactly one step further. And we take all the sap flows together, and then we get the change in water content. And then lastly, we need to solve the set of differential equations. And then firstly, we have the basic solvers, which are in Java available, which we won't be using for an issue I will tell you about later. And basically what you have in GROIMP is these two hard-coded functions. One is integrate and the other is get_rate. And these GROIMP knows that the integrate function always refers to a get_rate function, and the idea is that you put all your differential equations that need to be solved within this get_rate function. And this notation with like a double point apostrophe equal sign is the GROIMP notation for a differential equation. So we always start our get_rate function with defining the time it needs, over which period it needs to solve the equations. And that is like dt is equal to our period length. So for example, the entire equation set here is quite straightforward. These are implemented in our get_rate function. So for example, our osmotic potential, here I already replaced the R with like 8.31, so it's not confusing here. With then the sugar content, the water content for each plant organ. So we don't need to define, that's the usefulness of defining it as a plant organ. We don't need to define it for the internode, and then for the leaf, we can just use a plant organ. It's only for these specific things where you only have it on the internode. So for example, this summation of like from a leaf, you can never have water transport to various different plant organs. You only have it lost through transpiration. While for an internode, you can have it going to different plant organs altogether. And then there's an additional issue that if we want to solve this equation set, it is too complicated for the standard solvers in Java, because these are all non-stiff solvers. If you're not mathematicians like me, you might not know what a stiff problem is. So I added the Wikipedia definition, and it even says it's difficult to formulate a precise definition of stiffness, but it basically comes down to the fact that you have a system that is having some gradual changes, and it's very hard with a normal integrator to have a step size that is adequately small enough to solve the problem, because it becomes your step size becomes so small that it takes so much time to just solve the equations. And so what we need is a different type of solver that can adjust its step size based on the smoothness of the function that it's trying to solve. And luckily within Java, well, not within Java, there is this solver library called CVOD, which is exactly made for these types of problems. It's not written in Java, but there's a wrapper function in Java which is able to access these libraries. I've included these in the PowerPoint itself. So if you want the presentation afterwards, this is clickable. It contains all these files. And these are basically libraries written in, I think, C. And they can be accessed in GROIMP by using this code. It looks complicated, but it's basically just to add it to the path that Java knows where to find these solver functions. And then you can simply use GROIMP itself the same way you would use a normal solver in GROIMP. You can load the library with the function that I quickly showed in the last slide. And basically, you just make a new CVOD solver. You can set your tolerances of how much it can deviate from reality. And then you can basically just set the solver. And then that is, you can solve the equation set as I've shown it here. So let's take a look at some modeling results. Let's start with like a static example. I have here a soybean plant which has already been growing for a while that has the turgor-driven growth model implemented in it. And we start during the night, and I change the colors of our plant to represent the amount of turgor that is available in the plant organs themselves. And so red means that the turgor is decreasing below an equilibrium state. So it means that the plant organs are shrinking. It's losing water. And the high turgor, once it goes blue, it means that it's exerting pressure and the plant is actually able to grow. So this is mainly caused by the active sugar loading in plant organs that are still growing. So let's go over the steps. So now this is right before sunrise. Then the sun starts to shine. Our plants, our leaves, mainly at the top, start losing water through transpiration, and they start to lose water and also lose turgor pressure. We continue. Our plant is constantly losing water, and now we see near the night that already there's like a tiny bit of blue here in a newly growing plant organ, which is getting a lot of sugar, and the sugar is allowing it to create a positive turgor pressure and allows the plant to grow. And if we look at this system on a continuous basis, you can actually see the plant shrink during the day a bit. I can show, the next slide shows it a bit clearer, and then only expand during the night. So all the growth is taking place during the night, not during the day. And so I have a slide here that's showing with normal colors an entire simulation of like the turgor-driven growth in a soybean model. It takes a while to get started because it's much slower at the start. And so each second represents a day of growth. And you will start noticing that the plant grows like as if it has like a heartbeat. It's like it slows down and it even shrinks, and then it expands during the night.
How many time steps do you have in a day?
24. It's hourly. I solve it hourly. Yeah. You need to segment your day because the entire mechanism is like diurnal. So yeah, you really need your hourly time steps. I haven't tried. Then I need to change my sunlight model and probably the calculation time will also be a lot worse because this simulation, it stops now because GROIMP crashed. But I can do it on a supercomputer entirely, the entire growing season, and it takes like 24 hours for a plant like this to simulate the entire growing season. So it's quite a complex model to solve. That's for one. Yeah, but with for the lights simulation, I use the clones to get the shade. Yeah. Yeah, it is problematic to simulate multiple interacting plants, especially if you then add the soil component, because if you want to do competition for water, you actually need to have the soil component as well. So this is only the above ground.
No one question.
Yeah. I know you're working on it. And then I have a final slide about the sensors that you can use, because we're modeling all new types of processes and parameters basically. So one of the advantages of using this turgor-driven growth approach is obviously that you include the growth limitations based on water availability, but also that we're expressing growth in terms of quantifiable things. So the extensibility and elasticity, they are really properties of the cell wall of a plant. And also they are quantifiable processes like the sap flow, like how much how much water the plant is taking up during the day, but also the stem diameter variations or leaf thickness variations. So this expansion and contraction pattern is really measurable. It's really, you can really see it on an actual plant. And you can also measure the turgor pressure, but that's a little more, well, difficult or annoying. Because these first three, you can use continuous sensors. So you clip them on the plant and you just look during the day what the plant is doing. But turgor pressure is something you need to do destructively. So you need to take specific time samples. And if you really want the daily pattern, you need to start before the sun goes up and do it during the day. And it's also not that accurate, the ways to measure turgor pressure. But it gives you an idea of what it should be around, and you will really see the drops in turgor pressure during the day. So I've added like a graph here of 3 of the days of the soybean model, I think it is. And like the dynamics that you see, very basically, this is, I think it's the conceptual model still. The transpiration starts here at 6, but you see that like the sap flow, it has a delay, because the first water that is being used is the water that is already contained in the leaves and in the stem, and it takes a while for the water to start flowing and start refilling this water that is lost and to create this water potential gradient. And you also see that the water doesn't immediately stop flowing the moment the transpiration stops, because it also takes a while for the water that is lost to be refilled. So what are some of these sensors that we use in our lab? So at the top, you have the sap flow sensors, which we can basically clip around various plant stems. With some plants, it's easier than others just because of the amount of room you have available. For example, for a tomato plant, it's very easy. For a lettuce plant, it's almost impossible. But the basic principle on which these sensors are based is basically, you see also like insulation around this, is because they have a heater element that supplies heat to the plant stem, and then you have two basically resistance, one above and one below. And if there is no sap flow, like during the night, these resistances will measure the same temperature. But if there is sap flow, there will be more heat transported upwards, and you will have a higher temperature on the thermistor that is higher up than on the lower one. And then you can calibrate this basically by knowing the flow that you send through it, and you can really have these continuous graphs of how much plant the water takes up. And if you manage to connect a sensor like this on the bottommost segment of your plant stem, you basically know the water use of your entire plant during a day. And then secondly, you have these dendrometers, and that's, like I said, these expansion and contraction patterns. So basically, this is like a spring that is very, very sensitive, that can move forwards and backwards, that really measures how much a plant organ is shrinking and expanding. And if we look in detail on one of these sensors, so this is a plant equipped with both, like this is a tree equipped with a sap flow sensor, but also with a point dendrometer. And then you have, for the control plant, the pattern looks like this. So you really see every day the shrinking of like even woody tissue, the shrinkage every day. And then it gets refilled, but it also gets refilled more than what was originally lost. And that's your actual plant growth. So from this, you can distill what is the elastic component and what is the plastic, the actual growth component. And you also see, if you submit some plants to drought stress, that you immediately see like a deviation from this pattern, where growth entirely stops. Like we're not seeing any more refilling than what was lost. And then once you start irrigating again, you see a refill. That's sometimes like a growth spurt even to catch up. It doesn't catch up entirely, but it catches up a little bit with the other plants. And that was where I ended my presentation. If there are any questions, feel free to ask. And it's just 30 minutes without timing it. Yeah.
Thank you for this.
Yeah.
At the start, you mentioned that there was one problem with calculating water flow because it depended on water could only flow from connected internodes. And I was wondering the slope that you see at the start of the day for step flow to start up, is that dependent on that assumption? Is the
It's not really an assumption, or what do you mean?
Well, I mean, from the presentation, because that the step flow is calculated hourly, and within an hour, water can only flow from one internode to the next internode.
No, to every plant organ that's on top of it. So So that's why you need like the query, because it needs to take into account all the plant organs that are connected.
So then you're solving the equation. Trajectory.
And for stability reasons, it's better to only allow new plant organs to be formed at like midnight, because otherwise you change. Yeah, it's not handy. Yeah.
How does it work with the solver? Because if you have the time step of 1 hour, and you said that the solver can choose can adjust.
It can adjust the time. Yeah, I don't know. It's called like the backwards differentiation formula, but you can look it up. I don't know what it does. It is important to note that you basically have other interactions that you have to exclude. So, for example, I assume because there's like an interaction term between like the shrinkage during the day, I don't allow within the hour, as we're calculating how much the plant organ is shrinking, also recalculating how much it would capture light, because it's technically a slightly smaller plant. When I just calculated at the start of the hour, I calculate the transpiration, the light interaction, and then assume that is equal for the entire hour, because otherwise it would, I think it would never solve if you have to put the light model as well into the differential equation solver. I don't think it's gonna.
So the light calculations is done once per day.
Once per hour.
Once
Before, yeah. So you run the light calculation first to calculate the transpiration, and then you run all the differential equation. Yeah.
And then I was also wondering, so the way that a lot of FSPMs calculate growth of organs is through source sink relations.
You still need them.
Yeah, and am I correct in understanding that the source sink relation causes an osmotic potential in the leaves, and that causes the growth?
Yeah, in leaves and internodes. So basically, you still need to like use the sink functions to like determine what are your growing plant organs, and then you just divide all your sugars under your growing plant organs, and those are causing the osmotic potential drop, the turgor increase, and those are causing the growth. Yeah.
Any questions?
Go ahead.
A lot of them are related to scaling.
Yeah.
Let's start with your scaling. Can you elaborate a little bit on what you do with soil now?
We don't do anything with the soil, not in our lab. So we are basically assuming that the water potential we use as an input is like the water potential at the link between the roots and the stem itself. So it's already what the roots are taking up. So we're not taking resistances of the roots into account. I know what I saw in Berlin, that at least some people are working on the water flow in the roots as well, the interaction between roots and the soil, and that's an entirely different problem from this. Why? It's because now, like I showed the problem that your sap flow, your sap flow can only split up upwards, but in roots it comes together. So it's also like a different idea to solve, and I don't know if it translates perfectly with what the way I did it, but I haven't looked at roots yet. I guess you take the opposite approach, yeah.
So another reason I think the soil makes life very difficult is because the water resistance of soil is very, very nonlinear as it dries.
Yeah, the matrix potential as well, right?
Yeah. So how is that with the relationship between water content and your water potential?
In essence, you could simply combine both models if it's computationally feasible, and then you use a soil model and a root model just to calculate what the water potential would be on, like, from the root uptake, and then use that as the input here.
Sure. Yeah.
But then you probably need also like the water potential. Yeah, there will be some interaction, of course, because
now basically the question is scaling with a soil model is quite difficult because it's such very, very highly nonlinear. Are these nonlinearities as extreme in these plants where the resistances of the vessels decreases as rapidly as you see in soil matrices?
There are, like, we've only tested the model now. We don't go to like the wilting point, because then you do have processes where you have even irreversible damage. In trees it's called cavitation. It's like air bubbles forming, but we don't have these processes included.
Okay, so in the cases.
But that should have something to do with like how you would include that in the model is by making your resistance also like a mechanistic function based on like historic. It basically represents just like your resistance to water flow, and if you have cavitation or irreversible damage, that would impact mainly your resistance to.
That's what's currently not in there.
No.
Yeah, and then you mentioned that growing crashes.
No, but that was just for the visual part.
Yeah.
Well, it was on my laptop, not on my yeah.
we do want to use models, right?
Yeah, but I could simulate it. I just couldn't visualize it.
Okay.
I think it's mostly a RAM issue. I think if you can use like a supercomputer with like hundreds of gigabytes of RAM, it should be possible. But I don't think it's necessarily a computational issue. I think it depends. Because the thing is, why can we calculate a single plant? Because we don't have the soil interactions included in this model now. So it basically doesn't matter as long as we have the interaction of the transpiration through the light model with like the cloner nodes. We have the interaction in terms of water demand for transpiration, just not like plants taking each other's water in like a soil interacting. So in principle, yeah, you could have like I'm not sure. I haven't tried simulating multiple plants, like one being like starting off smaller than the other and having like less transpiration. I haven't tried to model multiple plants yet. Could be an idea, but I'm not sure. I couldn't really answer it.
Okay, because I think that's I think it's relevant that we start including this in our models. It's just a matter of can we?
Yeah, definitely.
Papers.
Who?
Richard Papers. He's also,
yeah.
He's been doing
Richard,
yeah. So we're thinking of how to do that scaling from the smaller scale step model to the larger scale growth models, and we can make some assumptions based on the behavior of the small scale model. Integrate that into the larger scale models is something we're interested in.
I must say this original model that I adapted it from, like the turgor-driven growth, was based on like simplifying the shape as well. So now we're using an FSPM where we want to calculate it in each plant organ, but basically the model was used for simulating a tree based on just this. This is a crown as just one block. This is the stem, and then you even have the lateral water transport, and this is the soil. And it also, like, if you can downscale your complexity of your actual structure, like, I don't know, having some heuristics of like, oh, these plant organs aren't changing that much anymore, we're gonna put them together in like one pipe and only focus on the growing organs. I think maybe you can look into simplifying for simulating multiple plants. Yeah.
Also the question on parameterization. You mentioned because sensibility, elasticity has been the main link between your water content and your potential. How easy are these to quantify on a spatial level? Variation is there on the spatial level? Is there? How sensitive is the system to these parameters?
Quite. Yeah, quite. So basically, you can mechanically actually measure these, but that's really hard. That you have to take tissue samples and stuff like that. But what we usually use to derive is basically calibrate based on these patterns. So we try to make the assumption that the elasticity for all the leaves is the same. For all the leaves, the extensibility is the same, and then we try to match this pattern. So match the daily contractions for the elasticity and the growth for the extensibility.
And you say it is quite sensitive to those parameters.
Yeah, but I think it's more to do with the fact that we want to make the assumption that the elasticity and extensibility are the same both over time. That's also an issue. Is it the same for a young plant organ as for an older plant organ, which is not the case because with an older plant organ. So we've done some simulations for grass leaves because they have a different growth pattern, because grass leaves have, for those who don't know, have a division stage, an elongation stage, and a mature stage. And it's basically like the cells are changing shape. And eventually you end up with a leaf that only has mature cells, and they've measured extensibility on these mature cells, and it's basically 0. So even if you create turgor pressure there, you will still see the elastic compression, but that's basically the mechanic that is stopping the growth. Even if you're adding sugar, is the extensibility going basically to 0.
I guess the extensibility is what sort of how we simulate the sourcing dynamics with a beta function.
Yeah. That's what I tried at first. So in this model, I did it as well, like even directly using the extensibility as like my sink function as well. So having like the plant, the organ age, like indeed having this function, like starting from, actually very high, because it's basically, the youngest plants are the most resilient, can grow the most, and then go down like with an inverse S-curve. But I found when I tried to calibrate this with my soybean model that this was not a good approximation, specifically for the start of the growth, because then you're creating a tiny plant organ with massive extensibility. It's taking up massive amounts of sugar with no like hard cap. It's basically what your sink function is then. And then it immediately, instead of having an S growth curve, it immediately shoots up. And your turgor shoots to like 5 megapascals. It's not realistic. So you need like the gradual increase. And it's probably like a combination of both. That's at the start, it's more driven by like cell division than actual turgor expansion. Like you have in the grass leaves. You have first the division and then the elongation on these.
And the variability between tissues and maybe more so between tissues and.
Sorry, what
the parameters, the sensibility, elasticity parameters, how variable it is.
I don't have the actual values for woody tissue, but they will be drastically different. Yeah. Definitely.
That's also
even for tree leaves, they will be drastically different from like crop leaves, because they're harder. But it's still measurable on tree leaves, the contraction patterns.
So basically parameterization is hard.
Yes. Yes. Especially also because if you want to do like a global calibration and you want to run this like very heavy model, like hundreds or even thousands of simulations, is very difficult. It's definitely the main limitation of using this model.
Okay.
And then the main advantage is that we can use continuous measurements that you don't have to.
You're at Google, and Google Go Lab has a virtual machine with 600 gigabytes, right?
Nice.
I think you should push a hand up for that. Julia had this machines.
Yeah, that, but there's also the solvers still need to be.
Yeah, but you can do the solver sequentially, basically.
Yeah, but the implementation of the solvers to the Julia.
Okay,
that's
yeah.
not yet,
Okay.
system. You need to connect
Yeah.
that solvers. That interaction.
Yeah.
I think that's.
Okay.
Sorry, my question is a bit too basic, but I was wondering, as you explained that it's quite computationally heavy to implement this process, and scaling has its challenges. So I was wondering, what is the main benefit actually of including this process in FSP?
So it's because if you want to really start looking at including your water limitations on your plant growth, you really need this, because you cannot just do that with photosynthesis. It's like in my earlier slides, you do have the closure of your stomata, so you have one response that you can add on your photosynthesis, but growth really slows down a lot more. So it's really, if you want to look at what's actually limiting growth, then you need to include this. So yeah, it's definitely depends on exactly what you want to do. Also, if you really want to study scientifically what is happening in the plant, then you can also use this. But it's definitely not for every application.
Maybe this is actually a good craft actually to demonstrate that. So the drop in growth in the red plant, and it comes back, and then there's a difference between the two. So that could be your stomatal closure.
Yeah.
But there's this period where growth hasn't happened. And
yeah. So basically,
been through a developmental stage, so there have been leaves that haven't been able to grow.
Yeah.
Depends.
Depends. Yeah, definitely yes. But we on the simulations and data that we found, for example, for maize, we found that development also slows down.
Oh,
based on loss of turgor. Yeah. But what you see here, like this growth spurt, is basically because photosynthesis slowed down less than growth, and it's adding like excessive sugars, and that's why it can do the growth spurt. Yeah. But yeah, sometimes even development is also linked to the turgor pressure. Yeah, otherwise you would see like you have a drought period in a maize field, and you would see like in the middle tiny leaves, and then again the long leaves. So it kind of makes sense.
Yeah, it's something that I'm sort of struggling with as well. Because theoretically, if turgor is like a thing on small scale, so it happens today or tomorrow, but if you look at longer scales, you can bank the sugars that it would make anyway. And just
up to a certain point. There's also like a negative feedback loop on photosynthesis, I think, from the amount of sugars that are stored in the leaf cells. There's also a feedback loop there.
But theoretically, if it would be able to bank it could just
it could basically
especially there's source limited models able to simulate growth over longer time spans quite well. So it seems quite feasible if that's the case. But then in relation to development, especially if you look at these smaller scale developments on smaller plants, not looking at trees, it makes sense that you want to know what your turgor is and then give it time, because your fruit development can be fucked. The leaf development can be disrupted in different shapes.
I was actually thinking about it because I wrote a proposal that I did in GAT. We were going to like FSPM for cucumber growth, and apparently if you have too much light, you have too little water potential, your fruit swings. The water just flows out because the whole water status of the cucumber, which is normally growing, is really dependent on the amount of sugars that you're pumping in there. That's why it expands, really. So same here, right? It expands because water is flowing in, you should put all the sugar in there. Then when you're just really high light during the day, cucumbers swings.
That's
the problem.
Yeah.
Like,
and if you have a process like that, you also need to adapt a tiny bit this model, because the way I showed you, it does not allow reverse flow, but it's easy to implement.
Yeah, I think in those particular cases for horticultural systems, for
most plants, there's no reverse flow, but there are like indeed from like fruits, and also we're doing some research on trees that apparently have foliar water uptake as well. Like they take up water from the leaves and that also flows into. So yeah.
Why is reverse flow not possible?
Well, it is possible, but the way I showed you, it's just like linear. Like I showed you, it can only flow into like the above lying things. And it would, I think it would make the solving substantially more complex as well. Maybe you should, yeah.
How do you limit the solver?
By predetermining if you're gonna have reverse flow or not, and not including the possibility at each possible. But I'm not sure if it.
Usually, if you don't allow for reverse flow, solvers tend to break down because if you do multiple time step thing, if it calculates that there is a large flow and you get to a negative water content, basically, then there should be able to be sort of reverse.
Yeah, but
so that they can then sort of
yeah.
estimate.
I've had no issues with the way I implemented it because I always checked that the sanity check of this model is checking if water in equals water out, basically. Like your transpiration is your water out, and yeah, they matched without like specifically allowing for
That could be because you're not pushing the limits of the
yeah, probably.
Because my experience, especially with these flows, can be very, very high, very nonlinear responses. And you don't necessarily you start with too large of a step size, which now could really be, if you're
could be
to be very, very dry, then I think you need some reverse flow or
yeah. Then you need a slightly different way of implementing it, but I think it's yeah, similar at least.
I see why you did it though.
Yeah,
it makes sense.
I think solver wise, it's going to be harder to solve. I'm not sure, but I think yeah.
See how exactly.
Because the reason why I say I think it's going to be more difficult is because if you look at my equations, what was it? Basically, we calculate the sap flow in a step. It's in my get rate function, but it's not a differential equation. So we can assume like we know it at this time step, but I'm not exactly sure. I should really just test it. It's always the easiest way to see if we can just do it in between those. Because it's here where we calculate the flow, we already like select during which plant organ.
Yeah, exactly, but you do have your. So you can get positive or negative flow depending on.
Yeah, but true. Yeah, it doesn't even. Yeah, you don't even need to implement it. It's automatically there.
So there.
Yeah.
So
okay.
The problem would be if you have that baseline potential minus your potential, right? So that allows flow to go to be either positive or negative.
Yeah.
And so if you were to put like a minimum function there, for example, say you
then I limit it.
you have to be larger than
True.
than one or can be lower than one, then you're going to break the system. So
So essentially, it's already
in
there.
It allows for backwards flow. It's just that you limit if you know beforehand that there's going to be backwards flow, so don't push them into the solver.
Yeah, makes sense.
That's wise, but yeah.
You said that initially the model was developing a very simple kind of structure that you're talking about the upscaling to FSPM. Have you tried both ways of different levels of complexity to figure out what is, let's say, the ideal level of complexity you need to get a set of good results?
Not really. Yeah, it's a good question, but now we haven't really retested it because usually we were focused on like making a model for a specific plant type, and then I made some simplifications. Like for example, on a soybean plant, they have like these 3 foliate leaves. So each leaf actually has 3 leaflets. That I did simplify. I made it like one function to not have like each individual petiole as well. And that did impact it a lot, but I didn't really have an optimal. I didn't really optimize the complexity of the structure. I think you could, yeah. The problem is every time it branches, you basically need to, you need to keep those individual segments there. And I think maybe if you can then identify like an entire stem fragment where nothing is changing, maybe you can put them together, but I didn't really optimize the structure for that. It barely worked well enough for my PhD, so I didn't have to optimize it.
Very technical question. What's the unit of your R in your rate function?
It's
Your resistance. So you just.
Oh, I should check the paper.
So your flow is in liters, I assume.
Yeah, resistance. No, it's in grams. The flow is in grams, and the resistance will be in megapascal per gram. So it's resistance, right?
That's not resistance.
Wait, hold on. I'll check the paper. Yeah.
It's close to maybe grams per second, right?
Yes.
Yeah, and yeah, it's there. There's an hour component there as well, but it's in my paper. My unit checkout. I'm pretty sure.
You get that value.
Yeah, you calibrate based on your model. Yeah, it's not something you measure.
So that's sort of esoteric abstract.
Yeah, because in my model, I also assumed it's like it depends on the length between my plant organs, which I take as the center of each plant organ. Also the thickness. So as the plant expands, I also make the resistance at the bottom smaller because we have more vessels.
So it's basically both a resistance factor that determines the flow.
Yeah,
but it's
also structural.
But it's also a conversion from potential to.
Yeah, it's It's basically in the pressure needed to push a gram of water over an hour, something like that.
Yeah, pressure needed to push a gram.
Yeah, so
okay, yeah.
Depending on how large your gradient is, it will be.
Want to get real precise, you can do silent visual measurements.
Yep.
And there's actually something called Buddha from Eindhoven made a non-destructive way of sound to measure silent vessel distributions.
But if you want to be really precise, you also have lateral flow from your phloem into your xylem. So yeah, and then you have flow in both directions, and then it becomes really fun. Well, both directions, both, and like lateral and vertical.
So we just calibrate.
Yeah.
If it wasn't hard, we wouldn't have a job. Yeah.
How many parameters?
It depends on what you assume beforehand. I'll just say it like that. If you look at my full soybean model, because you have like we also use like a complex photosynthesis model, which we calibrate separately and stuff like that. Kim Leith, like the combined Farquhar, Van Kameren, Berry model, which we calibrate based on like Lycor measurements, just like point measurements of the leaf, which is also a question if this is upscalable. This then you would need for like assuming that each organ type has identical like elasticity, extensibility curves. Then just per plant organ type, an elasticity, a shape of it as well, an extensibility, and also the resistance for your xylem. You will also need to calibrate like a base resistance that is then scaled with the distance and the basically like the area of the cross section of your yeah 3 per organ type basically. Yeah.
When you say organ type, because I'm not familiar with.
Just a leaf will be different from like an internode, something like that.
Each internode is a different organ.
No.
So
Like
internode
just
is one.
as an internode as a type. You can, and it's probably more accurate, but I think you'll be overfitting. Yeah, definitely. Yeah. Yes. Well, manually, you run a lot of you check first what realistic parameter values are. That's basically you determine a range, and then you make random sets of parameter values. You run like 100 simulations at the same time on a supercomputer, and then you check which are way off, which are better, and then it is a manual process still, but you can probably automate that.
And your random part is just less than hybrid square cube, less than hybrid cube or something.
I don't know anymore. Probably, yeah. It's been a while since I calibrated this. I'm not sure what I used to,
How can we calculate, if I remember correctly, for theoretical water question? Like the water that the plant, the maximum water the plant can uptake.
There's no real limitation to it. The limitation will be the maximum water that it will be able to take up will be equal to your transpiration, plus like a tiny bit of extra due to the sugars and the osmotic potential. But there's no like hard-coded limitation or something in the plant. Well, in the model at least.
So all the water it needs it can uptake.
Yeah, if you would in this model say, for example, the transpiration for this soybean plant is 1,000 liters this hour. Eventually, it will stabilize. But yeah, there's no like hard limit. It will only depend on your water potential gradient. So if you like, it's automatically quite within a normal range. But if you would artificially make this huge, and your resistance is small enough, you will also get it will refill in this model. And if you put your resistance very, very small, it will refill almost instantly. So it is really an important factor. So one of the things that you can check, like the differential between your transpiration and your sap flow will also, that's something you can determine the resistance from, how long it takes to refill.
Yeah, I think that's one of the things that the original looking into to sort of simplify the system between two sort of steady states. Whether the refilling speed is limited by the resistance of the pipes or the uptake rates was limited by the soil. Basically, those are the two limitations that we're looking at because that determines the steepness of the refilling.
Yeah, as well, but it's also like it's also with a water potential gradient, right?
Sure.
But more difficult. I know. But
yeah, but there's this clear sort of distinction between this the rate, the refilling rate shifts from being very steep to
okay.
When it goes.
Yeah, but because there are also there are physical limitations. That's maybe also what you were aiming at. Like I say, you can artificially create like huge gradients, but there are indeed physical limitations of how low a water potential can go before a cell collapses as well.
Yeah, sure.
Yeah.
This is much larger.
Yeah, a plant cell can irreversibly dry out as well. Yeah, we all know that. But it's yeah, but then you cannot refill anymore. That's a fact. Yeah, then your gradient doesn't matter anymore. The cell will, it's plasmolysis, I think it's called.
Yeah, but we're not necessarily interested in that stage, but just if it gets at some point in nature when it gets dry enough that the soil water resistance
increases it a lot.
Increases so much
Yeah.
that refilling is then limited by uptake rate rather
Yeah.
than transport rate.
Yeah.
And then so it's those two systems. Is there enough in the soil and then transport rate is limiting, or is there not enough in the soil and then the uptake rate?
Then basically then your grade, but still your gradient between your root, like the upper part of your root and your stem will be still quite small because it can, like the gradient between your soil and your root will be very large.
Yes.
And that's where the resistance is.
Yeah. It's it's a lot easier to solve that.
Yeah.
In better gradient between leaves and roots. Yeah. Basically, we don't want to go into a dynamic model where we have to do it hourly and then it goes into
Understandably.
if you want to use it in decades, that's really not.
Yeah.
Thank you.
All right.
Thank you. I think we can stop.
I'm gonna stay for lunch, but afterwards I'm gonna try to be ahead of traffic on the way home.